And like Richard said, I actually am a research scientist. I work in cancer. And so I'd like to title my talk to Gene Trinity and Beyond. How many of you guys know what a gene is? Oh, wow, a lot of you. That's fantastic. All right. Okay, so hold on to what you know about a gene, and we're going to get back to it in a second. I wanted to start off with a little bit of a review. Here are my jacket. So I wanted to start off with a review of basic cell biology because it's so important to understand cancer, and I want you to really know what I do. So the first thing you have to do is understand cell biology, and then once you know that, then we can talk about cancer. And then once we can talk about cancer, we can talk about what I do. So bear with me for a few seconds. And the most important thing for me and for you guys is just to understand the concepts. There's a lot of biology going on here today, and I don't want you to get bogged down in the details. Just hone in on what's interesting to you, and maybe you can take one or two things away. Okay, so the first thing I wanted to share with you is why should you care about cancer? And the reason for that is because cancer is a fatal and commonly diagnosed disease. And men, the most common type of cancer that's being diagnosed is in the prostate, followed by lung and colon. In women, the number one common type of cancer is breast, followed by lung and colon also. And then the most deadly type of cancer, I think this is pretty interesting, is lung, and that's for both men and women. And the number one way you can prevent lung cancer is by not smoking. So if you do smoke, or you know anyone that does smoke, stop. Because it's going to probably give you cancer, and it's a pretty horrible cancer to have, and it's not very curable. So just to start off with a little bit of a review about the levels of cellular organization, we're all made up of cells. Together our cells work to make tissues. These tissues build organs, and our organs work together with other types of organs to form systems. And these systems are then what forms us as an organism.
Be quiet.
This is what a cell looks like. I'm sure you guys have seen this all before. Yeah? Yeah? Okay. So just as a quick review, we've got the nucleus up here, followed by the rough endoplasmic reticulum, and that's just a fancy word for an organelle that manufactures our proteins. We've got some mitochondria. Mitochondria and the cell wall. And then I wanted to show you guys, actually, we'll do this first. I wanted to review the cell cycle with you because this is so important to understanding cancer. Cancer is an uncontrolled growth of cells. So in order to understand that, we have to understand how cells grow. During cell division, a cell has to replicate its DNA and then builds extra proteins, enough proteins to make a second cell before it can completely split. This process is highly regulated, and there are lots of checkpoints in here. You can think of it as a stop sign. The cell has to tell itself it's okay to go, I'm ready to go, or I'm not ready to go, I need to fix some of my DNA. And the process that I just spoke briefly about is the central dogma of genetics, the fact that we are all composed of our genes. And the way I like to think about this is starting off with the library. Let's say you visit a library, but you're not allowed to check out any of the books, but you really want to learn something new. What you're going to do is go find a book that you're interested in, take some notes, and then you'll use those notes to build the new project that you wanted to build. Maybe you want to build a dollhouse. So how do you liken this to what's going on inside your cells? Well, the library would be like the nucleus in the cell. The books inside of the library would be like your genes and your DNA. And then the notes that you take are kind of like RNA. They're not the exact same as the books, but they're close enough and they're good enough, and they take all the relevant information that you need to build your new project. And that project that you build are your proteins. So I just wanted to give you guys a view of what it's like inside a cell. This is what I think is a pretty cool video, and it's done by a group out of Harvard, and it's composed of and maybe you'll just get an idea as to how complex our cells really are. So what you're looking at is the inside of a blood vessel, and these balls that are rolling around are cells. That would be one cell attaching to the blood vessel. So this is a very active process, and you can see that it's using these proteins to attach itself. This is now the outside of the cell, and those big blobs are things that respond to signals in our blood. And then the cell has to take that information and bring it inside to where it can do something with it. So this is the process of the signaling coming inside of the cell. And you can see that this is just some of the structure that's used to keep your cells intact. And again, these are more structured proteins. Those ones are actually actin. I don't know if you've heard, has anyone heard of actin before? Yeah. You can just think of it as like, you know, the frame of the cells of your house. These are microtubules. They're very similar to actin. Again, they're used for structure, and you can see that they're meant to kind of move and come together. Now what you're looking at is actually that inside that ball is the signal, and then it has to move along the networks inside our cell to get to where it's interesting, and that would be towards the nucleus, which is kind of back there. And so actually what's happening now is those little strands are RNA that's coming out of the nucleus, and they're getting what we like to call translated into proteins. So there's the protein. And now these proteins can come together with other proteins to do something interesting. And here the proteins are coming towards the wall of the cell. And now they're getting placed on the outside wall of the cell, where the cell can use them as ways to talk to other cells.
Oh my God.
And so what you just saw was how a cell might respond to a stimulus, and so this is a white blood cell, and that stimulus is telling it needs to invade the tissue, and so that's what it just did. You guys like that? It requires genetic mutation, and this can happen randomly over a long period of time. You guys all know that you have 2 copies of each of your genes. So cancer requires you to lose ability of one of those genes, and then a second gene, of that same gene. So that's called the 2-hit hypothesis. So your first mutation is in what's called a proto-oncogene. That's a gene that pushes you into cell growth. And you can think about this like a car. You know, on a car you have your accelerator, your gas pedal. If your gas pedal gets shoved down, you're going to keep driving. But you have a way to override that, and that is putting on your brake or pulling in your emergency brake. And so those types of genes are called tumor suppressor genes. And so even if you get a genetic mutation in one of your oncogenes, one of your growth genes, you can still override it, and that doesn't always cause cancer. But what does cause cancer is when you lose your ability to the tumor suppressor genes. So again, like I wanted to say, is that you have to have mutations in 2 types of genes, and you have 2 copies of each of those genes. And so that's why you don't have cancer happening all the time in a body, but you are likely to have one mutation in either one of these genes in certain cells. But it takes a long time to accumulate enough of these mutations to cause cancer. So just to review, cancer is a disease of cells. It is caused by the uncontrolled growth of those cells, which can then spread to other organs. All cancer is caused by changes in DNA. Not every cell can cause cancer, and the reason for this is because our cells have a limited lifetime. They're programmed to die after a certain number of days, weeks, or even years. And so the organs that often are most susceptible to cancer are those that constantly have to regrow. So cells in our gut are treated with highly acidic stomach acid, and they have a short lifetime. And so those cells constantly have to undergo the cell cycle. And so those are the types of cells that are constantly at highest risk. In the United States, 1 in 2 men are affected by cancer in their lifetime. So it's very likely that someone you know or someone you may already know has cancer, and 1 in 3 women are affected by cancer in their lifetime. So now on to what I do. I use high-powered imaging to look at cancer. I use this is an image of my microscope, and it can image 200 microscope slides in about 12 hours, which is really fast. And what we do is we take very thin sections, about 1 cell thick, of tumors. I actually look at breast tumors, and we stain those with fluorescent dyes, and they give us markers on ways on how to look at the cell. So these are some images that I've taken, actually. This is all breast cancer. What you're looking at, the green is the outside of the cell. The blue would be the nucleus, and the pink would be this cancer protein that I'm looking at. So I'm not actually looking at genes, rather I'm looking at the output of those genes. I'm looking at their proteins. So you can see that there's a lot of red, and what that means is that there's a lot more of that protein that should actually be there, and that means that this patient is likely to benefit from a particular cancer drug. And here are just some more images.
Ew.
Ew, I think it's really beautiful.
No, I'm sorry.
You should see it when it's not big. That's cancer. Alright, this is actually lung cancer, and then this region. This is a necrotic or a dead region. And so what I do is I actually take these images and I count how bright. I can go back. I take these images and I count how bright my protein of interest is, and then we quantify that. We give it a score, and then we're able to determine how likely a patient is to benefit from certain treatment. So you can imagine that there are many genes in your body that will help, promote the cell cycle or stop it. And so that's the reason why we haven't found just one cure for cancer, is because everybody's cancer is a little bit different. It's not the exact same genes that are affected every time. And so we have to find ways to identify which genes are being affected in individual patients so we can correctly match the drug and the treatment to their cancer. So with that, I'm ready to take any questions, and then my email address is up here. If we don't get to your question, you're welcome to email me. Don't make any regrets. My email address up here. These are some great websites just about science in general. If you're interested, nobelprize.org. Semantics Inc. actually has great animations if you're just curious about learning more. And then radiolab.org is a great radio show that talks about all different aspects of science. And then there are some books too. So does anyone have any questions? I see one. No?
Right there.
Oh,
You, yeah, you. Yeah, you.
Do all cancers have names? Oh, that's a really great question. So I tried to touch on that a lot. The problem that makes cancer such a horrific disease is because it's not just one disease. It's not just one mutation that happens in everybody that causes the same disease. In fact, it's a lot of different mutations, many of which we can't even identify, that cause the same type of disease. So we are getting better at identifying the unique mutations in individuals, and that's helping us to match the right drugs to those individuals. Yes?
Have any of your family had cancer?
Yes, my grandmother died of breast cancer, which metastasized to her bones. That I think indicates that there may be heritable genetic linkage to it. So heritable means that it can be transmitted down family lineages. Some people are predisposed to having cancer. What that means is, if you guys remember the image that I showed of the two genes that you had, in some people, one of those genes are just born through a mutation. But they're not going to end up with cancer immediately because they still have to accumulate another mutation. Yes? On the way.
Can you name all the stages of a cell?
Can I name all the stages of a cell in the cell cycle? Is that what you mean? Yes. PMAT. Prophase, metaphase, anaphase, telophase. Cytokinesis. You got it. All right. How about you, Elizabeth? Yes.
If one is just a cell,
Can't you just excise the cells from a body? That's a great question. In fact, that's often the first treatment. The problem is usually we don't find cancer in a body until the cells have left their primary tumor and gone to a secondary site. The problem is we can't identify where that secondary site is, usually, until it starts growing another tumor. How is cancer mostly caused in women? So the most likely cancers in women, I think, we can go back to that slide. Perfect? Okay. So the most likely causes of cancer in women are breast, followed by lung and colon. Breast is often more curable because, like you mentioned before, you can excise the tumors, and so things like that, screen for early detection have eliminated a lot of deadly breast cancers before they metastasize. Yes. Is brain cancer curable? I know that there are treatments for brain cancer. We can actually look at the slide. I don't study brain cancer, so I don't want to give you any misinformation. But what we can look at is how deadly it is. And so it looks like, let's see it on here. So it looks like it's going to be, it's down in the bottom. So I know that there is treatment, but it's probably not the most deadly types of cancer. Yes?
2 minutes.
2 minutes. Any other questions? Yeah, all the questions. Okay.
Have you guys found a cure for any other cancers?
Have we found a cure for any other cancers? That's a great question. Actually, I'm so lucky to say that my teacher that I learned from at the University of San Diego in California, she discovered a protein that is now the target for a lymphoma. So that protein is called Abelson kinase. It's a signaling protein. And so that there's a drug that targets it and basically inactivates it. So if you have, controlled growth, you can actually stop the growth, and then eventually you can get the first cells to die off. So yes, you can cure some types of cancer.
Last question.
Last question. I see some girls in the back. If you can scream really loud. Okay. Right there, you got your hand up. Go for it. No! That's good. No.
waitresses will be here all week.